Okay, can you hear me in the back? All right, so I'm going to talk about the coolest problem in the entire world, which is mitonuclear epistasis, and try and draw some parallels between G by E and G by G. So if we look at complex traits like body height, this is a figure from Lewinton's book, somewhat politically incorrect figure, but nonetheless there's phenotypic variability here. There must be complex genetics behind that. And this is another figure from Lewinton's book, modified by Gould, which shows that there's a genotype space that develops into the phenotype, and of course the phenotype is what selection acts on, and therefore modifies the genotype. So we're interested in how these complex traits work and how selection acts on them. And the GWAS paradigm has been somewhat unsuccessful in many ways, trying to determine the genetic basis of that. And probably hundreds of millions of dollars have been spent identifying the SNPs that are responsible for these continuous traits. And this paper, this rather well-worn paper, has been cited maybe 5,000 or 6,000 times. The issue is if you look at heritability from parent offspring to heritability from GWAS, there's this big gap in the genetic plane and the phenotype plane. So people have wondered what is the source of this, and this missing heritability may be due to genotype by environment interactions or epistasis, which is a genotype by genotype interaction. So people have sort of tossed this responsibility onto those context-dependent problems that have plagued us for a long time. So just to put everyone on the same page, here's the famous Clausen, Keck, and Heisey study, just to understand G by E. We're sort of chopped off on the right here, but if you take a clone of a plant and grow it in one environment, you get a different phenotype if you grow it in another environment. And likewise, if you take a bad genotype and put it somewhere else, it can be good. So when a genotype differs in its phenotype in one place, you have a genotype effect. And of course, across environments, you can get a G and E effect. But when these norms of reactions cross, we call that genotype by environment interaction. And if you look at epistasis, it's in many ways the same thing. So epistasis is when the genotype of one at one gene modifies the phenotype from another gene. So if we have a nuclear locus that has different phenotypes, if you put those in a different mitochondrial background and they change, that could be a G and G. But again, when those norms of reaction cross, we call that epistasis. So there's an obvious sort of very simple PowerPoint version of the ways these things can be similar. So epistasis is a complicated problem. To really get at epistasis, if we had 20,000 genes in the genome, we would want to know the entire landscape of epistatic variation that is relevant to phenotypic variation. And that's a complicated problem. So the way we deal with this is to focus on, obviously, the most important section of this epistatic space, which is the mitonuclear part. And the reason for this is that mitochondrial function is important in basic metabolic processes. So if we look at the mitochondrial genes and nuclear genes that work in the mitochondria, or somehow have connection, we can sort of model mitonuclear epistasis as a subset of all epistasis and ask how that works. So that's our basic question, and the model we use to address this is Drosophila, and we've been using the Drosophila genetic reference panel to play with this problem. And the DGRP, as it's known, is a set of 200 or so strains that have been inbred for 20 generations, fully sequenced, transcriptional profiles made, a whole lot of phenotypes have been studied, and a lot of the community in flies and other even even medical folks are using flies to study this. And the trick that we play with this DGRP profile, so here's the core 40 as it's called, and they vary in starvation resistance from low to high. What we've done is use balancer chromosomes and other tricks to put melanogaster and simulans mitochondrial genotypes into a subset of these nuclear genotypes, and just made a row by column table of mito and nuclear genotypes. And the trick here is this is a home team, because melanogaster is the chromosomes. So melanogaster mitochondria are the home team, and simulans mitochondria are the away team. We're just curious in what the row and column and off-diagonal effects are in how these two things interact. So this panel took us about 2 years to make, and we're in the process of phenotyping a bunch of these lines. So the first thing we did was to look at development time as a sort of integrative fitness phenotype that involves metabolic processes. So that 72 panel genotype that I just described is also described here. So each of these panels is a nuclear genotype background, and within each panel are 6 mitochondrial DNAs that are placed onto that nuclear background. And the blue are the home team, melanogaster, and the red are the away team combinations. And if you look across this, you can see variation, and it looks sort of chaotic, but there's some interesting patterns. We would expect that the home team and the away team would be different, and we see that in these cases here. So here, the melanogaster mitos develop fast, and the simulans cause a delay, so that makes sense. But surprisingly, there's 2 out of the 12 are the only ones that actually show this in a sort of phylogenetic contrast. The rest of them show other sort of variable patterns. So here are a bunch of nuclear backgrounds that sort of see sign epistatic type variation. So for example, here is a mitochondrial genotype that has a delay in development. You take that same mitochondrial DNA and put it in another nuclear background, and it's accelerated. So that means there's a nuclear locus or loci that are talking to that mitochondrial DNA and altering its phenotype. So that is a nice example of an epistasis, a sign epistasis. And then of course we have nuclear genotypes that just don't care about the mitochondrial DNA. So these are sort of canalized, and there's very little variation across these mitochondrial genotypes. So there's all kinds of variability that you can see in this story, and we're interested in that, in mapping those traits. So that's G by G, and what we were interested in is whether the epistatic story could be modified by the environment. So what we did was take those genotypes and place them on a variety of diet environments. So here are 3 isocaloric diets: high protein, low carbo, equal, low protein, high carbohydrate, and this is the standard Bloomington diet, which actually is sort of here in this space. And we took these genotypes and placed them on these four diets. So this is the 6 by 12 mitonuclear genotypes, and this sort of tiling identifies the 4 diet environments that they were placed on. So this was a lot of work. This was 300,000 eggs and 200,000 adults. And we studied development time, again, on those various diets, and found that, again, there's lots of genotypic variation, and this G by G is itself modified by the environment. So this is a plot of that scaling from fast development to slow development, ranked in this order, and some interesting patterns come out. Here is the high protein diet that accelerates development, and low protein tends to delay development, and this is the cross-class correlation. And what we can find is that the role of epistasis is in fact enhanced in the more stressful, if you will, environment, which delays development. And whether that's a general pattern for all things, we don't know, but it's a very clear pattern that we came from this result. So the outcome of this is that, yes, environment does modify the epistatic interaction, and we can begin to sort of take this apart. So let me just show you the thing that is the suggestive title here. So if we take a couple of these and look at them, here is this co-adaptation nuclear backgrounds that disrupt the co-adaptation, and that's a form of sort of phylogenetic epistasis. This black line is these same data, and the other lines are the different diets. And what this shows is that this is nicely epistatic, but when you change the diet, the epistasis goes away, and that's sort of interesting. Here it's this sign epistasis case, and if you look at that, the black line is the same data, but at different diets, that epistasis is modified. So that's sort of a visual version of what I think is going on. Okay, so this is an example where the diet or the environment, in a sense, eliminates an epistasis. So we're in the process of doing RNA-seq on those diet mitonuclear combinations, and we have currently some data on this problem from RNA-seq from a different environment, so I'm shifting gears a little bit, but we're using the same concept. And we're going to ask, what does the hypoxic environment do to mitonuclear gene expression? So we're going to shift to looking at gene expression in this thing. And we're using hypoxia because the reason you breathe oxygen is because of complex 4 that accepts the electrons and allows the mitochondria to work. So we believe the oxygen environment is a really important environment, if you will, for the mitochondria. So what we did was take a 2x2 table of these genotypes. So this is melanogaster from Austria, melanogaster, the standard Oregon R genotype. And then we put those nuclear backgrounds onto 2 mitochondrial genotype backgrounds and made this panel of 4. We've reported these genotypes in other papers, put them in a hypoxia chamber over a time course and froze them, and then did RNA-seq on them. And the experimental design is a really simple factorial: 2 sexes, 2 nuclear backgrounds, 2 mitochondrial genotypes, and a time course of hypoxia in this chamber. And then you do RNA-seq, and to make a very long story short, we found all kinds of genes that are modified by the genotypes and modified by hypoxia, and the females and males differ considerably. One interesting outcome is that the males seem to not do as much as the females, which is sort of counter to the Frank and Hurst idea. And This is a multidimensional scaling story. And interestingly, the females show a very clear genotype-specific response. So there's this paradigm of the hypoxic response, but what we're showing you is that each genotype has its own hypoxic response, and that's a challenge to sort of basic medical studies. But here's a case where in the males, this nuclear background, the mitochondria don't matter. In this nuclear background, the mitochondria do matter in their hypoxic response. So again, this is a G by G by E interaction. So here's the simple question I want to ask. How do we determine if G by G and G by E have common elements? So one way to do this is to fit a model in our gene expression data of genotype, hypoxia, and genotype by hypoxia interaction, and ask what those genes are. Then take the same data set and fit a model where you have mitochondrial genotype, nuclear genotype, and look for the mitonuclear epistatic genes, and then write those down. And the question is, the null hypothesis is if G by G and G by E are independent, then those loci will be not the same loci. And the alternative, of course, is that if G by G genes and G by E genes are the same, if they're 100% overlapping, they are the same genes. If there's some overlap to those, we can test them. So here's what we found, and I wouldn't have this title if we didn't think there was something going on. So if you take the G by E genes, there were about 800 of them, 900 of them that responded, these mitonuclear genotypes responded to hypoxia. And fully a quarter of them were also genes that were epistatic genes. And there's some asymmetry here. That is, if you take the epistatic genes across the data set, there were 1,800 or so, and maybe 14% of them were epistatic or were G by E genes. So this is certainly more, this is 4 or 5 times more than you expect by chance, and maybe 3 times more than you expect by chance. But some of the genes that are epistatic are indeed those genes that are mitonuclear. Here is an example. Here's an Oregon-R, mitonuclear. It doesn't really care about hypoxia. Here's a gene that, if you take these other genotypes and put them across this in a hypoxic environment, they very much care. And this is an example of this G by E type genotype. So here's the idea. If we have G by E gene, it's a gene that is altered by the external environment. A G by G gene is a gene that is altered by the action of another gene. And I'm just arguing that this other gene alters the cellular environment. And if the external environment and the cellular environment are connected in a pathway, then G by E genes can be G by G genes. And this is a simple way, I think, to connect genotype by environment complexity with epistasis complexity and mechanistically go and see if we can find what those genes are. So thanks for listening, and I'd like to acknowledge Jim Mossman, a postdoc who did a lot of the work. Leanne was here at the meeting, and 3 grad students who also here. I'm happy to take questions if there's still time.
Great talk, David. It seemed like there might be more mtDNA-mediated plasticity in males than females, assuming the overlap.
Right. And in a sense, what there is in this particular set of nuclear backgrounds, there's more mitonuclear epistasis in males than there is in females. But there isn't systematically more mitochondrial effect in males than females. So that this Frankenhurst idea absolutely relates to that and changes it somewhat.